I'm going to take on some of the clinical trials aspects in cure. It's a very big field, so I'm just going to focus on a few areas with the time we've got available. So we'll talk about a rough framework for the ways in which cure is being tackled. We'll go through the patients with the London and Dusseldorf patient in Tim Brown as well, and I'll pick on a couple of sort of high-profile studies in the field with broadly neutralizing antibodies and some combination intervention studies that have been reported recently. So I think whenever we think about ways in which we can control viral replication of ART or even try and purge or eliminate residual HIV, as all described by Denise, there's two main ways to think about it. One, we increase the power of the immune system to attack where HIV is and control it, or we do things to push HIV down or away. So that is the left side of the little seesaw there. So we could do latency reversal to remove residual virus or lock that virus down with lock and block. And then the other main interventions are all really trying to provoke the cytotoxic CD8 positive T cell for an increased HIV-specific immune response. There's a lot of focus on that CD8 T cell that's normally designed to go and get viral infections. Well, how can we make that go and attack or control HIV more effectively? And you can see some of the interventions that are being studied there. And the middle of the seesaw is just to talk about ideas in which you could make cells resistant to HIV infection, which really is by altering CCR5 expression, which is the cure patients to this point. So as a little bit of background, there have been some high-profile cases which are summarised, some of them which are summarised in this little figure. And you can see that this is the idea of when do people rebound after they stop ART. Denise described the acute infected cohort, and typically the median is about 2 weeks after stopping ART you'd rebound. But there have been these other cases, so the Boston patients who received bone marrow transplants and then they stopped ART and took many months to rebound with very low reservoirs. The Mississippi child took over 2 years. Baby was infected at birth, received ART as soon as it was infected, and then stopped ART and rebounded. And then there's Tim Brown, and post-treatment control is a separate issue, which I think Eugene might mention. So why these cure cases or these remission cases, it really does come down to understanding about CCR5. So you can see in the figure here, the idea is that this is the CD4 T cell on the bottom part of the figure, and the top is the virus, and this is the envelope spike coming out of the HIV virus. And this is the envelope trimer, is the first part of contact with the CD4 T cell, and you can see that it has to require to contact CD4, and then it has to also contact this other receptor, CCR5, or less commonly CXCR4. And then some people just naturally don't express CCR5, and the people who don't express that are what's called homozygous for this delta 32 or 32 base pair deletion for CCR5. That's about 1% of Europeans, and there's a little bit of evidence that it's actually possibly deleterious in long-term life. But in general, it's always thought to be essentially a safe phenomena to be not lacking this CCR5. But if you don't have it, HIV would not be able to enter those CD4 T cells. And this is the basis for why these bone marrow transplants have worked. This is Tim Brown. This is his oncologist who had the idea of giving him this bone marrow transplant that did not express this CCR5. This is Tim at the 10-year anniversary. He's 12 years on now. And this is the summary of Tim's case from the New England Journal. So essentially, they always have these figures which sort of show the viral load, where the treatment was given, where the treatment was stopped, and the nature of the chemotherapy. So as you can see here, he had leukemia, and he actually had to receive 2 bone marrow transplants. He was extremely unwell, and this is his viral load. He actually stopped ART around the time of the first transplant and then had another brief period and then stopped when he had his first bone marrow transplant. Excuse me. And then since then he remained off ART with undetectable viral load, and he had immune response after that. What's really interesting about Tim Brown, he actually takes antivirals still. So he was on antiretroviral therapy, 1 pill a day, had his bone marrow transplant and is considered cured of HIV, and now he takes PrEP. So the really cynical people, could say he still takes that pill a day, even though he's cured of HIV, but obviously he's concerned about not becoming infected. This was at CROI this year. There was an enormous amount of press around this London patient, which was sort of widely published in the media, some of the news outlets that published it, and it was presented in a conference and at the same time published. So this gentleman and the other patient in Dusseldorf are in a cohort in Europe called ICI-STEM, where they are recruiting individuals with HIV who have hematological malignancies, and then following them through the process of transplant and trying to understand the factors which lead to whether they control HIV or not. And the common feature amongst Tim Brown and the two subsequent cases I'm presenting is that the new bone marrow they get from the new donor does not express this CCR5, and it is now thought that's the basis for why these people maintain long-term suppression of ART. So he was a man, was diagnosed off ART for a long time and was commenced on ART soon after he was diagnosed with malignancy. He had a fairly close match. He had fairly standard conditioning chemotherapy, and as you can see here, this was his viral load. This is the period of ART. And you can see here, this is when he receives the bone marrow transplant. And what this orange line is, when you say 100% chimerism, means he's completely adopted the new immune system that doesn't express CCR5. So any residual virus in his body would be unable to infect those new CD4 T cells in his body. He had actually a very stable course, was only in hospital for 1 month, and had some mild graft versus host disease. I've sort of mentioned these key components, but basically he adopted this immune system which didn't express CCR5, and there was a range of experiments done that showed that these cells from his new immune system couldn't be infected with R5 protecting HIV, and he's been extensively studied with multiple samples, not of tissue it should be said, but extensive sampling of blood, and there's essentially no evidence of HIV evident in this man. So he's now been off ART for 18 months as of CROI, which is March of this year. And the reason why this was probably such an important finding is that it showed that Tim Brown was probably not an anomaly. Maybe Tim was just a one-off, but this is a very similar sort of event, and it just shows the significance of this CCR5 disruption. And there are other strategies where people's T cells are being taken off, the CCR5 is being removed and being reinfused. So this will be continued to be pursued, and it's really important to understand that this can't be done with HIV routinely. The mortality from a bone marrow transplant in this setting is about 1 in 3. So it really is not an intervention that can be performed widely. And then also at the same conference was the Dusseldorf, again, in this man from the same cohort from Germany. A similar story. He received this, he had leukemia, had a bone marrow transplant. The new donor didn't express CCR5, got slightly different clinical course, had some relapse which required further treatment, and had graft versus host disease. And this is his diagram that shows all that there. Essentially, it's hard to see, the viral load is listed as the blue line down here, the aqua sort of blue line at the bottom. And this, he's gone through his transplant here where the red bag is. He's developed this chimerism, this 100% chimerism, so he's adopted this new immune system of the donor, and he's actually only stopped ART way out here. So when this was presented, he'd only been off ART a short time, 3 months, so it's too early to say if this man really is a true remission, but clearly it's very interesting. It's similar to the previous patients. He was extensively sampled as well, including tissue assays, and again, essentially no virus could be found. And this is very hopeful that this will be another patient who is going to have long-term remission of ART. And this is the rough summary of the patients, and I suppose the biggest point is here is that Tim Brown's been off ART 12 years, which is the ultimate test. London patient 18 months, and the Dusseldorf 3 months. There's a lot of similarities in how this has occurred. The other really fascinating paper, which I'd recommend, 2 pages of really interesting reading, was only published a couple of months ago, and this just shows what we can do with data registries. So in Britain, they have death registries, and they have, if you want to volunteer yourself for having being a potential donor for a bone marrow transplant, you have a registry where the sequences of your genotype are kept. So there's up to 400,000 people on this registry, and they can also follow those individuals over time and see if they died. And essentially they looked at individuals to see if you didn't have that delta 32, so you were homozygous, therefore didn't express CCR5, would that have any impact on your life? There's not people with HIV, there's not people, with getting transplants, there's just people in the community in the UK. And essentially, this one little figure shows that if you've got this delta 32 mutation homozygous, your likelihood of reaching age 76 is lower than if you didn't have that in the general population. And there's been other reports that if you don't have this delta 32, if you have this delta 32 deletion, you're at higher risk of mortality for things like influenza, and West Nile virus has also been reported. So this is really interesting, we always think of the positive benefits of this delta 32 homozygous deletion and what benefits that could have for people with HIV, but actually this is this idea that you don't understand fully all the deleterious effects when there's manipulations to the genome. So it's just a fascinating use of this registry data and understanding that when we're doing things, we've got to think about other off-target effects and long-term effects. So now we'll go on to talk about broadly neutralizing antibodies, because there's so many interventions. I've just got to pick on a couple, and there's been a lot of interest in these antibodies. For example, Pedro mentioned them yesterday for treatment. They're being studied for prevention in a very large trial in Africa, the AMP trial. But I'm looking at them from sort of this cure perspective. And essentially, these people, everyone with HIV develops an antibody response. They attempt to neutralise the HIV virus, bind to it and lead to control of the infection, but it's largely ineffective. But some people have been shown that they develop these antibodies and they have this ability to bind to HIV and control the viral infection, or be able to effectively stop it reinfecting new cells. And I'll show in a little figure that it's not just that, but there's a lot of other potential benefits of these antibodies. And they've been identified in individuals, and then they've been able to be cloned and then produced in large quantities in the same way as a monoclonal antibody, so then they can be produced for clinical use in clinical studies. This is an example of, this figure here really shows the light blue is the HIV, so this is the membrane of the virus, and this is that envelope trimer in light blue in the background that I showed before with CCR5, for example. And all of these coloured blobs are little, the FAB binding sequence. If an antibody's a Y, it's the little act that binds the antigen bit on the end, and that's the spot where they're binding on the envelope spike. And the little names there are all the names of the antibodies that are now being used. Many of these are now in clinical trials, and you can see that it's not just one spot. There's antibodies that can penetrate and go into that envelope and bind that envelope spike in different spots. And the envelope spike has this shield around it. It's called a glycan shield, and that's the reason why the normal immune response for people with HIV, it's very difficult to get in there and bind effectively. But these antibodies have this very special attributes where they can penetrate that glycan shield and they can get in there and bind to envelope. And these are the things that they can do. So the first thought about why they're effective is just this little number 2 here. That's the antibody there binding to the virus, and when it binds to the virus, it neutralises it and stops it infecting the cell. So it stops this idea that it can go in and make new versions of itself. But in addition to that neutralisation potential, it has these other effects that brings in other immune cells. So, for example, it binds to the antibody, the virus. It's presented to the immune system by dendritic cells, and this then stimulates the immune, and then the dendritic cells chew up the virus and present it to the immune system, and you can get this. You potentially can get this potent viral response by CD8 T cells, and it also can potentially lead other cells like these killer cells and these macrophages to come in and recognise viral infected cells and eliminate them. So that's where a lot of the excitement is with these antibodies in the cure space. This is a trial that had a lot of profile late last year and early this year, and it was giving a combination of these antibodies to people with HIV and then stopping their antiretroviral therapy. So these are 2 of the antibodies that are currently being used. Again, a confusing picture, but all I'm just trying to show you is that the blue one is called 101074, the red 3BNC117, and they're binding to this HIV envelope spike here as this little squiggly pink and brown, and they're doing it in 2 different spots. And their ability to attack it from 2 spots, the rationale was that it would lead to increased potency. This is how the trial was performed. You can see that up the top here, people were on antiretroviral therapy, then they stopped it. ATI means analytical treatment interruption. They received 3 doses of these antibodies at high levels, and then they watched these individuals over time, and they saw when their HIV virus rebounded. And you can imagine if you think these antibodies are acting like a vaccine, or they're having some wonderful immune response, that they could control that viral infection over time. And this is the main sort of findings of the paper. Essentially, viral rebound was delayed for up to 21 weeks. I won't get into all the details of the sensitivity testing, but if the individuals were sensitive to the antibodies, there is some testing that needs to be done for that. That rebound was about 21 weeks. And by and large, it's when the red and blue lines you can see in these little individual patient graphs, when the antibodies disappear from the system, by and large, the virus does come back over time. But there were a couple of participants which were of great interest. You can see this is the levels of the antibodies over time. The black line is the viral load down the bottom, And these individuals maintained viral suppression even though the antibodies had disappeared from their system. So the idea was that, was this just a natural phenomenon? They were always going to control viral replication anyway, or was it something about these antibodies that were controlling the viral replication? There's been a subsequent presentation separate of the paper which has shown one of these individuals actually has had rebound of virus at approximately 1 year, and one person is still under follow-up off antiretroviral therapy for a year without rebound. So again, these could be natural post-treatment controllers, but it's also very interesting that maybe this is an immune effect of these antibodies for such prolonged suppression. And this is another thing that's only presented in a plenary session. This paper hasn't been presented as yet, but this is really exciting area, and there's a real debate on whether these antibodies do induce these increased CD8 T cell responses or increased HIV-specific immune responses. And there is a debate about this. We wait for this paper with interest, but this is people on this trial who were virologically suppressed, so they didn't have viral load up in their blood stimulating an immune response towards HIV, but they developed increased HIV-specific immune responses under the influence of these antibodies. So the idea is that these antibodies are taking virus, presenting it to their immune system, and leading to this increased HIV-specific immune response, a bit like a vaccine. And that's why this term vaccinal effect of these antibodies is being applied. So watch that space. If you ever see a presentation on these bNAbs and they talk about them increasing immune responses, could be a very interesting debate, and we'll see more data with time. But this group looks like they're going to publish on this, and they've postulated this before, other groups have not. So in the time I've got left, I'll take you through another. So these bNAbs, there's been sort of 3 or 4 presented in clinical trials, and then they're just going to keep coming harder and harder and harder. And these antibodies are going to be presented in cure sessions, they're going to be presented in clinical trial sessions, and they're going to be presented in treatment sessions. So getting your head around bNAbs is going to be useful down the track. So the reason why I'm presenting now, this is the glazed over moment, because this is a monkey study, and everyone goes, oh, the monkeys, don't talk about monkeys, I want to talk about people, we're clinicians, but these studies are being done in humans already. These antibodies and both these interventions have been adopted by Gilead, so these are effectively going to be agents that are trialled in clinical trials and going to be presented and taken on by pharma as well. So this is a monkey study, and we talked about bNAbs, and essentially TLR agonists are complicated and they have multiple effects, but if you just think of them on the seesaw on the right side, increasing your immune responses towards infections or viral infections, and they also have an effect to increase those cytotoxic T cell responses. So they're sort of a multi-pronged attack on HIV to increase those immune responses. So this was looking at basically whether this combination could lead to increased likelihood of viral control. As I mentioned, these drugs are also in other studies. The name of this one is vesatolimod, which is always confusing, but if you think about toll-like receptors, they've always got toll within these names. They're immune modifiers, they're called IMODs. So if you see tolimods, they're going to be TLR agonists, and there's a range of different TLR agonists. Gilead is presenting their first trial at IAS, a dose escalation study in people with HIV, and there's another trial that's been underway for some time. This antibody is, as I said, has just been presented at CROI, some data, which if I've got time, I'll flick through, and it's being investigated by other investigator-initiated groups as well. So this is the study. Essentially, these are monkeys that are, and really importantly, they're infected, and only a week after they're infected, they're put on suppressive antiretroviral therapy. So this is the classic example why monkey studies, everyone, their eyes glaze over. This is not real world. People don't go to an ART within a week of being infected, but this is a very controlled environment. And there's essentially 4 arms to the study. One, monkeys get nothing. One, monkeys get 1 intervention. Another arm gets 1 intervention, and the other arm gets both the interventions down the bottom. And they're kept on antiretroviral therapy and suppressed, and these interventions are only given while they're suppressed, and then well after these drugs are gone from their system, they stop ART a couple of years down the track. They then took lots of samples of these monkeys, and they showed that there was no evidence of these drugs around before they stopped ART, and they actually showed that they really couldn't find HIV in these monkeys before they stopped ART as well. And this is the main result of the paper. And essentially, these are the monkeys that got no intervention. They all rebound high viral loads, which monkeys do very quickly. And then if you look at the combination of the 6 of them rebounded, so 5 of the monkeys had no rebound at all, and 1 intervention, 1 stayed suppressed, and one of the antibody intervention, 2 stayed suppressed. So 5 monkeys effectively have been cured. And this is the ultimate way it's done in monkey science, is that they take samples from the monkey. This is pretty full on. Lymph nodes and lots of blood, and they give that to other monkeys like intravenously. It's the ultimate way to see if you could infect another animal, and they show that they did that and they could not infect other animals. So this adoptive transfer process was also successful and sort of evidence of cure of these original monkeys. I mentioned this vaccinal effect. Did these things increase this HIV-specific CD8 T cell response? Are they acting like the vaccine? And in these monkey studies, they showed that was not the case. They did not lead to that increased response as a reason for why these monkeys were cured. But this is probably really important. The monkeys that didn't rebound, they were just infected. They had very low viral loads and very low reservoirs before they were put on ART. So it's sort of like this sort of slightly false scenario of these animal studies. And these monkeys were the monkeys that didn't rebound or were cured. So they had very, very low reservoirs and very low viral loads. That's only sort of 1,000 copies was their peak viral load before starting ART. But it's an important study, proof of concept, which is what these studies are trying to do. And maybe if these interventions can be combined, you could lead to control of HIV infection. And these studies are now underway with these combinations and other combinations of antibodies and toll-like receptors. So watch for these combination in studies that are of immune interventions in the future. And here's an example of them. As I said, when you see the GS, right, in front of the number, you know that it's Gilead Sciences. So you know that they've engineered this antibody that was called PhD-121, and it's now become a drug for study in clinical trials from pharma. And they're combining with TLR7, and there's also a therapeutic vaccine that Gilead have acquired, which they're using. So we need to just, again, watch this space. The first studies of this antibody then were also given in people with HIV, and this was presented at CROI. From what I can understand, this hasn't been published as yet. But similar to what I'm going to show you from the study with the combination antibodies that led to suppression, some of these studies led to suppression as well. So this antibody, PhD-121, and this is, you can see the little figure here. This is the people getting the DIN. It's dose escalation. They're getting different doses of the antibody. And some of the people didn't have HIV for safety, and then some of the people did have HIV. And some had lower viral loads and some had higher viral loads. I'm just going to present the 2 people that are sort of interest. And again, these are not, so it's always important, these are not controlled studies, these are not randomized studies. You know, these could be one-off events and things. But in this field, there's a lot of studies with these small sample sizes reporting on these sort of individuals. So again, the idea that individuals with relatively low viral loads are given a single infusion, these people are off ART, of the antibody. You can see this is the red is the drug level disappearing there, and then you can see the virus is also undetectable on a standard assay, and the antibody's gone from the system and the viral load stays suppressed, and this person then popped up their viral load. So same idea that some individuals might be getting a longer term benefit even after the antibody has left their system. And that's all we've got. There wasn't a lot more discussion. It was put on the end of the presentation. Interestingly, that presentation was done in a treatment session. That wasn't done in a cure session. It was actually presented along with ART trials, and that was considered to be a treatment intervention. So I've just got a minute or 2 left, and I'll just talk about one study at the end, and this kick and kill, which was quite high profile, but it was completely negative. The intervention had no effect, is the short story. And this is a sort of a study that had been awaited for some time. It was the River study out of a group in the UK based in Oxford. And this was a sort of a classic kick and kill where they were giving a therapeutic vaccine. So this idea that you give a vaccine to someone with HIV on ART, that'd lead to increased immune responses, and then at the same time you'd give those individuals a drug to turn on viral transcription or increase expression of virus or viral proteins, so then that increased immune response can come along and identify those infected cells and they can eliminate them. So that was the whole kick and kill. So this was a classic kick and kill trial that was done there, and it used a therapeutic vaccine. You can see there this Chad 63 MVA, and the latency reversal agent was vorinostat. And for the sake of time, I'll just flick through the design. It was a randomised study to give individuals just ART or to give them the combination of the vaccines and vorinostat, and the outcome of the study was just looking at the reservoir. So the measures that Denise had discussed, they just looked at the amount of HIV DNA left in these individuals after this intervention. Again, I'm on time, so essentially these were men, and you can see 30-odd people in each arm, 60 people in the study, who were all virologically suppressed, and their CD4 counts were excellent. And this is the ultimate money, is that the blue dots represent the people who just had ART, and the red dots just represent the people who got the whole intervention, and this is looking at the amount of HIV DNA left in their bodies. And you can pretty easily see that at any time point along there, there was no difference in the reservoir from this study. And they had different ways of looking at this. This is, Denise described, the viral outgrowth assay, a very sensitive assay. Again, no difference in the two arms. So a negative study. There was some thought that there was this increased HIV-specific CD8 T-cell response, and maybe there was an increased immune response towards HIV, but there was no effect of that seen on residual HIV in those individuals. The study's not published as yet that I'm aware of. So it was a highlight. The importance of this study was that it was an important kick and kill study. It was safe. The CD8 T-cell responses were driven, but it didn't lead to a reduction in the reservoir as expected. So I think I'm on time. I've got through, and I'm going to stop there. Thank you very much.